everyone. Thank you for joining us today for our Medical Pathways webinar, sponsored by the Claude Moore Charitable Foundation, where we're all going to be learning more about health sciences. My name is Rachel George, and I will be your host for this evening. CEE, also known as the Center for Excellence in Education, is a nonprofit organization founded in 1983 by Joanne P. DeGennaro and the late Admiral H.G. Rickover, who is the father of the nuclear navy and of civilian uses of nuclear power. CEE's mission is to nurture high school and university students to careers of excellence and leadership in STEM. You may visit CEE's website to learn more about the center's cost-free programs for students and teachers, the Research Science Institute, the USA Bio-Olympiad, the Teacher Enrichment Program, and STEM Lyceums. I am pleased to have three wonderful speakers for today's event. We have RSI alum Katie Hoover, who works as a clinical systems director. A little about her. Katie leads international teams to implement and manage cloud-based applications. The systems house critical documentation and data for highly regulated clinical trials. With more than 20 years of experience in the biotechnology industry, Katie has a breadth of experience from drug discovery and analytical chemistry development to digital systems process and clinical studies. She earned a BS in biology and a BA in English from Virginia Tech. Our second presenter is Dr. Jasmine George. She is currently the manager of advocacy programs for Research America, where she accepted the Louis Sullivan Fellowship. Jasmine received her BS in biology from North Carolina A&T State University, and both her MS and PhD from the Morehouse School of Medicine in Atlanta, Georgia, where she was a RISE Fellow. Her thesis work included clinical studies and community outreach in underserved populations. Jasmine previously worked as a quality control lab tech for Noramco Inc., a former subsidiary of Johnson & Johnson. She is actively involved in diversity, equity, and inclusion for the National Science Policy Network, and is also the co-founder and co-creator of the HBCU Science Policy College Tour, which aims to increase minority access to science policy and science communication resources. Finally, we have Miss Meredith Lewis, a former Claude Moore scholar who grew up in Loudoun County, Virginia. She graduated from Tuscarora High School and studied at Radford University's School of Nursing. Meredith currently works in the medical intensive care unit at Duke University Hospital. In her free time, Meredith enjoys reading, hiking, and playing with Loki, her Australian Shepherd. Thank you, ladies. I will now turn it over to Katie Hoover, who will share about her STEM experience, what she does, and the opportunities offered in her field.
Hi, everyone. So I'm here to talk to you about the career opportunities in biotechnology. It's also pharmaceutical companies, but mostly drug development. Thanks for joining today. I'm Katie Hoover. I'm the Director of Clinical Systems at NeoImmuneTech, Inc. It's in Rockville, Maryland. Although I live in Portland, Oregon, and I work remote most of the time, but sometimes I go into the office in Maryland. I work on strategic leadership, project management, process improvements. I'm also a business systems owner, and I'm focused on data integrity. And this is all within the context of drug development. If you'd like to contact me after, you can always look me up on LinkedIn, and here's my contact information there. I just want to talk a little bit about some influential college courses. I know most of you are in high school, and so I had a little bit of an unusual made some unusual choices in my college career here. So I was a Bachelor of Sciences in biology. I was really focused and loved science, but Virginia Tech is a great and very large school. So I noticed that I was in these really large classes, but I really wanted to talk to people about art and life. So I got a Bachelor of Arts in English as well. And here are some of my favorite classes that I focused on. I'm also a creative writer. And I realized that part of being a scientist is also communicating about science. And that's something that is always needed. So I started to really enjoy classes like technical writing and figures and tables for scientific publications where you can really hone in on the exact message that you want to tell in a picture with as few words as possible. I also really enjoyed an English class called Ecology and Literature, which brought, how science and our environment affects us as people. And I just found that to be really important to make sure that we understand that we're all involved in science every single day just by living. The next thing is I joined the cooperative education program. It was at Burroughs Wellcome at the time. Burroughs Wellcome was in Research Triangle Park in North Carolina. And now when I started, it was Burroughs Wellcome, and then it became Glaxo Wellcome, and now is known as GlaxoSmithKline or GSK, one of the biggest pharmaceutical companies in the world. I worked in the Department of Drug Metabolism and Pharmacokinetics there. I started when I was 19. It was a great experience. I'll talk a little bit more about that. I also did a study abroad at the Estación Biológica La Suerte in Costa Rica, and I studied ecology and conservation of the rainforest. So this is what Burroughs Wellcome looked like, I think, when it was first built. 1969, this building was built. It was called the Spaceship. It was a very important architect that designed the building, and so it was a great place to work. At 19, here's me and my little badge, and and the unicorn that I identified with very much at the time. And so this, it was a great place to be. And just a little bit more about cooperative education and why I chose to do that. It gives you great experience. I'm a type of person that likes to do something for a reason. It was hard for me to apply what I was learning in the classroom sometimes to real life. And I really liked working with real life problems and understanding why I was doing it. So the experience and the on-the-job training of cooperative education was very important for me. Also, Burroughs Wellcome was already large at the time. They had a great cooperative education program. And so I met a lot of other aspiring college students that wanted to be scientists as well. So the community was great. There were people from Virginia Tech and Penn State, both there in the community. And also I didn't receive a lot of financial support from my family. So the paycheck was really important to me. And it was a great experience overall. While I was there, I worked at night so that I could also attend the study abroad in Costa Rica. I waitressed, which I'm really bad at waitressing, so I will never do that again. I already decided a long time ago, but I did go to study abroad in Costa Rica because I thought maybe I want to be in drug development, or maybe I want to be a research scientist in the rainforest. So I checked out this place, and it was an amazing experience as well. We sat in the rainforest and observed for 6 weeks and wrote a paper about, for me, I wrote a paper about the behavior of northern jacanas, which is a marsh bird. It was an amazing experience. Here's me walking across a log in the rainforest. This here is a palm tree, a walking palm tree, and it moves throughout the rainforest over time. It's like searching for nutrients, so it will send roots down into the ground, and the ones behind it will die off because they've already used up all the nutrients there. And so over time, this tree walks through the forest, which I found to be really fascinating. So when you are going through college and you're having all these new experiences, I really encourage you to look inside of yourself and ask yourself why. Find your why. What is the thing that motivates you the most in your life, and what are your purposes? For me, I know that I like to ease suffering, I promote inclusion, and I like to simplify complexity. I really enjoy these things, and I think they all kind of go back to easing suffering. Challenges come up, and when you're able to step back and say, why am I staying up really late doing this? Or why am I doing a task that I don't enjoy? And then to go back and say, because like, for example, in drug development, I know that even if I'm right now, checking 1,000 pieces of information for accuracy. I know that, that may not be something that I enjoy doing, but I know that in the end, I'm working with a team that's going to bring critical treatments to patients over time. And that's really important to me. So just a quick overview of the drug development process. This shows a bunch of pills at the top and calls them compounds. Obviously, drug ideas don't start as pills. They start as ideas, or they start as, peptides or proteins or monoclonal antibodies that eventually become, packaged in a pill in some cases. But for this purpose, there could be 10,000 compounds that you will see at the beginning, and then it moves into sort of a preclinical development where, you select just that amount of molecules that you can test deeply enough to see if they have promise. And then you start to move into clinical development, which means in the clinic, which means having to do with humans. And there are phases of those tests. So there's the effect on the body, safety in humans, effectiveness at treating diseases, large scale safety and effectiveness. So this is a smaller scale version. Is this effective on just a few, like 20 patients? And then you start to go into larger groups of people, and that's where you'll get more data and information about the effectiveness and safety. And then, of course, the long-term safety. And then after all of that, you may come up with one compound that is approved as a drug, and maybe you don't. At the end of the day, it's still, up for discussion with the regulatory authorities. And then one thing I want to point out about this whole thing is that every single step of the way, there's data that needs to be protected, and that the integrity of it, so that it cannot be changed, is very important. This is very generic, but just to give you a sense of how long this takes. Basic science is 2 to 5 years. The preclinical testing is 1 to 2 years. Clinical trials testing in humans is 5 to 7 years, and then government approval 1 to 2 years, and then you have an approved drug that can be marketed and taken by patients. So for me, these are my drug development personal experiences. I'm going to read through them, and if you guys have questions, you can ask me later. So for drug discovery, I folded and characterized synthetic proteins for a novel drug discovery platform. For drug development, I characterized drug stability and degradation, for example, shelf life and toxicity of degradants. And I also designed chemical tests to verify results of drug manufacturing steps. I also, as I was talking about the DMPK, so that is drug metabolism and pharmacokinetics, administering drugs to animal models, and then measuring the concentration in the body of those animals in different organs given at a given time point. So I may take, at the beginning, I may not have any of that drug in my body, and then I take the drug, and then it goes, does it go into my stomach after 10 minutes? Is there anything in my kidney? Is there anything in my brain or my blood? And then after an hour, testing those same organs to see where that medicinal compound goes in your body. And now I work in clinical studies, and I'm responsible for the cloud application that houses critical documentation and data and tells the complete story of a clinical trial. And so just thinking about clinical studies, drug registration is more like regulatory. And each country has a specific criteria for drug registration. And those health authorities will look into this cloud application that I'm working on to understand everything about the clinical trial. And so these health authorities include the FDA for the United States. For Europe, it's EMEA. For Canada, it's Health Canada. And for Japan, it's PMDA. And then I just want to show you how chemists celebrate Thanksgiving. So when I was folding proteins, these are a few of my colleagues. I want to talk just a little bit about LinkedIn because I was able to contact both of these old colleagues from back, in the early 2000s to ask them if it was okay if I used their picture in this presentation. They were very happy to say yes. And so just, chemists, you're always wearing protective gear. And even when we're celebrating Thanksgiving, so here's me in my goggles, and then my friend Tomasz was deep frying a turkey for the team, and outside he had on his face shield and his lab coat, and then our fire extinguisher here. We were very, very hungry chemists, and this is my friend Jason. And yeah, it's just safety first and the way to celebrate Thanksgiving. Just a really high level clinical trial management. While I was developing drugs and working in the laboratory, clinical trials seemed very far away, and it took me a long time to understand, really anything about clinical trial management. If you are interested in it, I have this book, which you can also get online, of course. It's the FDA Good Clinical Practice. And, there's so many regulations, and don't let it daunt you because it's, there's a lot of information out there to help people get involved in clinical trials. But just at a very high level, the sponsor is the owner of the company, the owner of the intellectual property and the drug. The clinical research organization is the team and company that helps the sponsor to run the trials and make sure that they comply with everything here. The principal investigator is the main doctor that is influencing the trial and being an advocate for their patients. And then each site is a hospital or a clinic where the subjects or the patients go, and each site has a doctor that runs that site and is responsible. And here you see a doctor and her patient possibly talking about new drugs that can help in the future. The trial master file is what I work on for the most part right now. These are some zones of places where I collect information from all of these different tests and many different sources of documentation and data. I don't want to go into that unless you guys are interested in it. This is my last slide. I just want to tell you that the career research tools that have helped me the most, LinkedIn is by far the best way to get in contact with people in the drug development and biotechnology and pharma. There's a lot of networking that you can do there. There are tons of job listings and also skill learning. Glassdoor, you can do company research here. You can also see job listings and you can easily apply. Then there's salary.com. And it helps you see and set expectations about what types of salaries you may want, what types of salary you can expect for a certain job type, or if you want to negotiate your salary with research, you can use salary.com. And of course, the company of interest, go to their website and just read all about them and what jobs are available. And then I just want to wish you all the best. And this is another one of my whys. This is my puppy, Obi. And this is right when he was learning how to walk on a leash. And he was very happy and proud on this day. Thank you very much.
Thank you, Katie. I love the Thanksgiving photos. I got a kick out of them.
Good.
So that was a really great presentation. If any of you have any questions for Ms. Hoover, feel free to put them in the Q&A function. Now I'm going to turn it over to Dr. Jasmine George.
Okay, so I'm Dr. Jasmine George, and I'm going to talk to you a little bit about my career path today. So currently, I'm a manager of advocacy programs at Research America. but I am a recent graduate actually in my PhD in biomedical sciences. So I'm super excited to talk about my journey. And of course, if you would like to reach out to me, you can find me on LinkedIn under Jasmine George, PhD, or my email is on the side here, jgeorge@researchamerica.org. So, how many of you are familiar with this career path? You go to college, you go to professional school, you make the money. Yeah. But what if I told you you can become a scientist at any point in time during this path? So after you go to college, you can decide maybe graduate school or professional school is not for me, and you can teach school, work in industry, work in government, or work in a nonprofit. You could decide to go to a master's degree or a postbac experience being external research experience, and do the same thing. You could obviously go to the professional school, get your postdoctoral experience, and then go into the same thing. So what I'm trying to tell you here is that there are many ways to do science. There's a bunch of different arrows on this, so it's probably going to pop up. So this is the way that I do science. We're going to talk a little bit about my journey and how I got to manager of advocacy programs from science. So I am from Virginia Beach, Virginia. That's that corner of Virginia here, right on the water. I grew up 5 minutes away from the ocean, but I can't swim. So don't contest me on that. I decided to go to school in North Carolina. And then after school in North Carolina, I decided to pursue my graduate studies in school in Atlanta, Georgia. Took a gap year in between my PhD in Athens, working at Johnson & Johnson, and then moved right back to Atlanta to pursue my PhD or doctoral degree. So these are a few pictures throughout my life. Here is my PhD graduation, that's the most recent one. Here's me at Johnson & Johnson. You can't really see the lab as much in the back because it was a very secret lab. So you couldn't get too many pictures, but I did get one in the corner here. Here's my first ever white coat ceremony. I was so proud of myself standing up on that stage and saying, I'm going to be a doctor someday. And then finally, one of my proud moments, meeting the late John Lewis when I did a Hill Day or experience in Washington, D.C., talking about insulin prices. So this is kind of my linear journey, but it's not so linear. When I was where you are, I wondered what my next steps after high school would be. So my entire family had gone and received education from historically Black colleges and universities, going to places like Norfolk State University, Hampton University, and North Carolina A&T. So I set my sights on North Carolina A&T because at the time it was the #1 producer of Black scientists and engineers, and my aunt went there. So at first, I wanted my major to be psychology because I wanted to go to medical school and become a neurosurgeon. So looking for opportunities in medicine at school, I stumbled upon research. So here's a few pictures for you guys. I had to go through my MySpace for this, so I hope you're happy. Here is my dorm room at North Carolina A&T. Here is me posing with the welcoming international students to North Carolina A&T. I was a welcomer, greeter, and I helped students, learn English and things. And so here's the North Carolina A&T sign, and finally a program from when I joined the Beta Biological Honor Society. So as I said before, I kind of stumbled into research, and research allowed me to step out of my comfort zone. So my first research project was in bioinformatics, which is a field where you use computational tools to interpret biological data. And for my bioinformatics project, I looked at proteins that could link to arthritis in mice. And that was an interesting experience and kind of one of my first formative experiences into biology. And from then, I switched my major to biology, and I knew that I wanted to just pursue that biology/research path through school, with a focus, hopefully, of going back into medicine at medical school. So you could see how my ambitions kind of grew. So my next out of the comfort zone experience was my second research project, where I traveled on a plane for the first time ever, going to Michigan State University. And I did studies on the evolution and survival of a flower species. So I spent a lot of time outdoors. This is me outdoors with a tiny baby turtle. And it was a very formative and good experience, and it really did push me towards liking research even more. And then finally, my most formative and longest project in undergrad was in biomedical science, which is the area where biology and medicine meet. And for that, I spent 1 year doing research on enzyme degrading of the kidney in diabetic kidney disease. So all of these experiences honed my research interest to biomedical science. And so I spent my last 2 years applying to master's degree programs. So I chose graduate school because I knew that I did not want to focus on becoming a practicing physician anymore. I wanted to go into research, and I wanted to have my own lab. And so in order to do that, I knew I needed to go to graduate school, but I felt like I did not have enough experiences in research to, make it into a doctoral program right out of undergrad. So I decided to do a master's degree. And during my master's degree, it ended up being the right choice because I learned a lot of lessons. I learned how to change my study skills. I learned new time management skills that I never thought that I needed. I of course learned new research techniques, but I also here learned to fail. In fact, I'm going to clue you in on a little secret. I failed one of my classes in my master's program. I was shocked and devastated because up until that time, I had never failed anything before. And, sitting back, I was like, am I ready to become a doctor? Is this the indicator that I should not do this career path? But through that failure, I also learned resilience, because I learned to pick myself up, take that class again, and go on to do both clinical wet lab research and some community outreach, completing my master's degree in time. So after I completed my master's, I started looking towards doctoral programs. Unfortunately, this first go around, I got denied from every single PhD program I wanted to get into. Couldn't decide why. And so I decided that I wanted to find a job and reapply. So I applied everywhere and finally landed a job with Johnson & Johnson as a quality control technician. So quality control is a field within industry where scientists test the purity and safety of drugs before they reach the consumer. So the job took me even more out of my comfort zone, because now I had to go from my biological research lab mindset into an analytical chemistry one. And that was a very formative experience, I would say, that helped focus me in the future. And I still use some of my principles and things that I've learned from industry to this day. So after spending my year in industry, I finally got into a couple PhD programs, and I decided to return to Morehouse School of Medicine, where I spent 6 years publishing 7 original research publications and receiving 3 research communication awards. And this is one of my original research publications in the Frontiers in Immunology, which I was extremely proud of. So what did I do for 6 years? I made mice fat. I made them very, very fat. So I studied lipotoxicity, or how fats can become toxic in diabetic kidney disease. So we fed mice a high-fat diet, modeled after the American diet, and let them get really obese. And these were special mice that were predisposed to high blood pressure. So to briefly describe this picture, I used mice genetically predisposed to high blood pressure to observe how a high-fat diet affects kidney outcomes over time. So on the left, you see a mouse kidney under a microscope with a normal diet and without disease. And on the right, you see a mouse kidney on the high predisposed to high blood pressure, fed a high-fat diet with disease. Very different looking kidney structures there. So coming to the PhD program at Morehouse, I still had a goal of pursuing research in my own lab. However, curiosity took over when I began an interest in policy. So I went to a Hill Day, or opportunity for PhD students like myself to interact with members of Congress, and was confronted with another side of research and reality. So I realized that there needed to be people who led the charge for important social and science issues, because the social issues will eventually affect the science issues. So coming back from that trip, I started to look for more ways to get involved. And I quickly noticed that a lot of the policy, health policy opportunities and science policy opportunities were relegated specifically to medical students. And so there I found a gap for opportunity. And I developed the Morehouse School of Medicine Health and Science Policy Group, an inclusive and interdisciplinary group for health and science policy. This kind of gap seemed to extend throughout the HBCU ecosystem. And so myself and one of my very best friends came together and developed the Historically Black College and University Science Policy College Tour, which is focused on giving students, much like myself, who are curious about science policy, opportunity to gain some experience through workshops and engagement opportunities. That set me on fire. So I knew from then on, I'm done with being in the lab. I've been in the lab at this point for almost 10 years in between undergrad and my doctoral program. And I decided that I wanted to do policy full time. So I pivoted once more, as I always do, and I found opportunities within policy. And that led me to Research America. I started at Research America as a Lewis Sullivan Science Policy Fellow, using that opportunity to gain invaluable firsthand experience in advocacy. And then they liked me so much that they hired me to be their manager of advocacy programs. So there, I'm continuing to grow my knowledge and develop some skills in project management. So who is Lewis Sullivan? Just to back up a little bit. So for those who aren't familiar, Dr. Lewis Sullivan is a health policy leader, minority health advocate, author, physician, and educator. He served as the Secretary of the United States Department of Health and Human Services during the Reagan administration, and is the founding dean and first president of Morehouse School of Medicine. His legacy at MSM and beyond was one of the reasons why I applied to the fellowship. So through the fellowship, I was able to accomplish so many things. I wrote for the Research America blog. I met with top officials to talk about workplace diversity, and I completed a day one project for the Federation of American Scientists, proposing collaborations between the NIH and the Department of Education to bolster the biomedical research workforce pipeline. So I'm looking to create more opportunities for students just like you to get into biomedical science just like me. And lastly, I want to show you that your science can be used for many things. In addition to communicating my science in poster form, as seen as in pictures throughout this presentation, I've turned my science into comedy. So here's a picture of myself and several other scientists around the Georgia area who did science comedy events for a nonprofit called Science for Georgia. So this is one of the many ways you can actually just make your research super fun and become engaged with anybody who will listen and make them laugh. So what do I want to do? What do you want to do? Here are some examples. You could go into industry, You could go into science policy. You could go into academia. You could go into government. There's so many ways that you can do your science. And finally, my advice to you all. So I'm a big sister, and so I'm accustomed to giving out unsolicited advice. So I will advise you all to ask for help, okay? There is no shame in asking for help. Find your mentors. So find a person who you connect with who is doing the things that you want to do and talk to them. Because let them mentor you, let them guide you. Ask questions. You will have the opportunity later to ask questions, and I'm hoping you'll take my advice on that, because if your question is answered, then you have more knowledge. So ask some questions and think big. Remember the fact that you are the future of science and science policy or whatever field that you go into. And so think and dream big. Thank you all.
Thank you so much, Jasmine. What a great presentation. I really appreciate you opening up about your resilience, because that is very true to your accomplishments through and through.
Hello, everybody. My name is Meredith. I am a registered nurse, so I get to take the lovely work that these two other ladies are doing and kind of get the chance to, after years of research, apply it in real world practices. Like I said earlier, I am from Loudoun County, Virginia. I grew up here, and then I had the opportunity to, in high school, attend the Claude Moore Scholarship Program, and I started as a junior in high school, attending a program that was at the time named the Introduction to Health and Medical Sciences, where I got to shadow different kinds of healthcare from a podiatrist to memory care to emergency rooms, and learning about different kinds of career within medicine. I got to have some background knowledge in anatomy and physiology and medical terminology. And I decided from there that I wanted to go into nursing. Up until that point, I had been more interested in science and research, but for myself and the aptitude and interest that I had, I found that I was going to be more successful in a hands-on field. I had a problem sitting still, so I knew that I needed to be on my feet, and I could combine my interest and love for science in a way that I could also be working with my hands in a day-to-day while working with people. And I fell in love with nursing through that. So I had the opportunity to, in high school, begin a nursing program and kind of get a feel for if that was something that I wanted to do, thanks to the Claude Moore Scholars Foundation through the Academies of Loudoun providing that chance for children and young high schoolers in STEM. And from there, I realized that that was something that I wanted to pursue. And I applied to college, and I got into Radford University, where I later joined their School of Nursing, and I graduated in 2021. I don't have as much career experience as the other panelists here, but I graduated with my bachelor's in the science of nursing. There, I got to participate in all kinds of things, and I learned that there's a lot that you can do within the career of nursing, whether that is nursing research. You can do nursing. There are all different kinds of nursing, whether it's bedside, you don't have to deal with patients if you don't want to. If you want to have the holistic model that is nursing, it comes from a different perspective of medicine and looking at the medical model and how you handle patients. The nursing model looks more at holistic care than disease process, and I found that very interesting. It just takes a different approach to looking at the body, and it allows for there to be, in my opinion, a multifaceted approach to make sure that people get taken care of in the best possible way. And that really spoke to me as I discovered kind of the paths I wanted to go down, and being more interested in critical care medicine and the body and the disease processes that patients go through when they are very critically ill, to be able to incorporate the holistic model that nursing provides to care for them in multiple facets. While I was there, I had lots of opportunities in the clinical setting as well as research. I believe the middle picture was through the local health department in 2020 giving out COVID vaccines. That was a pretty cool opportunity for me, that after these drugs were developed, to be on kind of a front lines of administering something that I feel like I'm going to look back on historically, be extremely grateful that I got the opportunity to participate in something like that. So currently, and after I graduated from Radford, I applied for a job and I started working in the medical intensive care unit at Duke University Hospital. And the patient population I deal with is anything from advanced respiratory distress syndrome, asthma, acute asthma exacerbation, GI bleeds, acute kidney disease, acute liver injury, chronic cirrhosis, a lot of sepsis, things like that. And then taking patients at pre-organ transplant when they're being worked up. And we get a lot of rare things. And then I just get to help people. I don't have a lot of pictures, unfortunately, of what I do day to day, because I can't really take pictures of that. But I've had the opportunity to just over the past about 2 years, learn a lot and grow, and then also had the opportunity to participate in some research opportunities, as I believe Katie was talking about, when it gets to patient clinical trials. We do a lot of study drug research and tracking patient symptoms. Kind of as the study drug is administered, we get to be the eyes that help you look at what kind of side effect after it's passed a lot of those rounds and gets to be used on real life patients. So that's been pretty cool opportunities that I've gotten to have. And then since then, I have just loved that nursing has provided me the opportunity to always be continuously learning. Since I've started, I've continued to get certifications in my field, and there's always something to advance to. And I have enjoyed the mobility that nursing has allowed. There's a ton of job opportunities, and I'm always learning about new ones that aren't always what you would think of with nursing, on both, like I said earlier, the inpatient and not dealing directly with patients. I just love being able to help people, and I love that I found a way that I can combine the kind of critical thinking aspect of my brain that can break down things, and then the part that likes to see the outcome that people have. It was a way for me to satisfy the scientific and inquisitive part of my brain that's always asking questions, with kind of also instant gratification and being able to hands-on deal with people and see the impacts that research and that our actions have on them. I don't have as long of a career history as the other wonderful people here today, but I would be more than happy to answer any questions about how I got to where I am now. And as I continue on, because we're always advancing and always striving. So I think that's all I have for our question section.
Thank you so much, Meredith. What you said about being the eyes, it's a great illustration of your position. So thank you so much for your presentation. And that, once again, everyone, was critical care registered nurse Meredith. Now is our question portion of this webinar. So I know we have one question so far, and I'll just read this out loud. So Henrietta had asked, where do 10,000 compounds at the beginning of the drug discovery process come from? And Katie Hoover had answered, molecule libraries created by service companies, synthetic peptides used as inhibitors, molecules that have in the past been proven to have some therapeutic effect. New ideas from you. And Henrietta also asked another question. Dr. George, can you tell us more about science and comedy? And then Dr. George responded, I have done improv comedy where we pretended to be molecules, and I have done stand-up comedy sets at Dragon Con and at the zoo. So those two very good questions. And so please feel free to ask some more. An anonymous attendee has stated, what do each of your typical workdays look like? So starting with you, Meredith, what does your typical workday look like?
Yes, so I work 12-hour shifts. So I kind of start my day. I get report on my 1 to 2 patients, just depending on how critically ill they are. And I start my day. I will do my assessment, kind of going all through the body, if they're on what IV medications they're receiving, if they're on a ventilator, what their ventilator settings are, if they're on dialysis, both continual dialysis, if their kidneys have taken a real hit versus intermittent, things like that, and just seeing where my patients are at. And then we do interdisciplinary rounding where I work. So are either advanced practice providers such as nurse practitioners, PAs, our attending physicians, our respiratory therapists, all kind of collaborate to talk about the patient, and they'll go through their vital signs, if there's anything abnormal in their assessment, their current medications, and their pertinent lab values, and then make every day, we make a new plan that builds on the last plan, because especially in critical care medicine, things change more by the minute than by the day. So you're always kind of reassessing and making a new plan every day. And then from the plan that we make, usually I have a lot of orders to carry out. So whether that be drawing labs, implementing certain hanging medications, changing if we're changing anything up, adjusting some of our more life support machines. And then I go through that. Basically, my day is broken into like 3 sections, because I do 3 assessments throughout the day. And then we trend lab values. I manage all the medications. And then a respiratory therapist manages the ventilator, but any other life support measures that the patient is on is typically my responsibility to manage, as well as kind of being the first eyes to look at any lab values or medications, and noticing any abnormal lab values that need to be escalated up to providers. And then also there's a saying in nursing that it's like, you're the last line before a patient receives a medication. So always questioning things, not so to say, just because there is a medication ordered, you always need to make sure that it makes sense and you're the one questioning it, because you're the one administering it and you're like a safety line for that patient. And then I go through with my 3 assessments at 8, noon, and 12, getting labs along the way. And then I do anything from assisting with bedside procedures and like lumbar punctures, inserting of invasive lines, things like that. And then transporting patients if they need imaging and things like that. And then mainly being an advocate is another big role of nursing, being a patient advocate. You're the one that sits with them all day. You're the one that talks to their family the most and talks to the patient the most and sees them change throughout the day. You're basically constantly assessing and noticing any changes, and then being the one to advocate for whether we should or shouldn't be doing something. And then I end my 12 hours giving a report to the next person, because nursing is a 24-hour job. So that's kind of my day to day.
Perfect. Thank you. Dr. George, what's your day to day?
Oh, not quite as intense there as yours. No, that's I commend you, really. I very much so admire your work, and yeah. So my job, I usually do a check-in with the full team early in the morning. Then it depends on if I have a briefing or something going on at the Hill, because Research America is an advocacy alliance. And so we pay very close attention to what's going on in Congress, and legislatively, and in science. And so if there is let's say that Congress is having some kind of meeting, I'll probably, log on or go onto the virtual platform and attend. Then I take a lot of calls. So a large part of my job is creating programming around advocacy. So I take a lot of calls, I talk to a lot of people about their concerns, and I also help around with like writing for the blog. I'll write, I've written a policy memo, so policy memos get written all the time. And Research America is an alliance, alliance. So we represent multiple different entities in our organization. And so we usually have at least one alliance discussion per week, which is just a 1-hour meeting where where our member will talk, or we'll have like a speaker come in and talk. And so lots of meetings, lots of writing, lots of paperwork.
Well, thank you. And Katie, what is your day-to-day like?
Well, it's varied. That's one of the things that I really love about my job is that it changes a lot. I have long-term projects, but also on a day-to-day, something can come up in an application that needs to be solved right away. But in general, the company that I work for is a smaller company. We have, a few studies. We have 7 active studies right now. And so I'll join into the team meetings for each study, not all the time, but many times, just to see, what's happening with the study, keeping tabs on it to make sure where the applications that I am responsible for can support those team members in a better way. Also, let's see, this morning I attended a meeting, a seminar, a webinar about archiving requirements by the EMA and how that's different in the United States and how it's also different between good clinical practice and good laboratory practice. And so I learned these things from regulators or people that have long-term experience, and then I apply them to my company, which is a smaller company that's just building up some of its processes. So that is, let's see, so it's more of a startup company that I'm working in. If I were working in a much larger company, which I have done in the past as well, some of them have been around for literally 100 years. And so like Abbott Vascular I worked at, and they had been around for 100 years. So in that case, instead of like creating the new processes that we need to follow, it would be, bringing people from using paper into digitizing their work. And how do you transition from a process of using paper all the time to always using electronic means of communication and storage. So those are just a few of the things that I work on a day-to-day basis. Oh, and I wanted to say, Meredith, that a lot of people, after they have worked very long time lifting people and working with them directly, they move into clinical research because that a lot about, how to care for the patient, and something that somebody that's more of an administrator doesn't know. So, hey, when you're ready, we're here.
Perfect. Thank you, Katie. While I have you, it looks like we have another question for you. When looking at the drug discovery process, do simulations play a role?
Definitely, there will be, simulations, but I'm not sure exactly which simulations you're talking about. Is there an example of a simulation? Maybe a model, like a model, like a animal model?
Sure, let's go with that.
Okay, so, definitely, preclinical studies are often done in an animal model, and the hope is that the animal will reflect the way that a human will react to a similar drug. And so in those cases, sometimes it does, that an animal model can simulate the effect of the drug on a human, and sometimes it doesn't actually do that. So there is more of an effort in the recent years to move from animal models to more computerized models, and that even though that effort is being made, it still needs to be accepted by the FDA and by the health authorities. And so this will be, definitely something that I hope that some of you will work on.
Perfect. Thank you. I'll also read out the last question that our anonymous attendee had written. On average, what patient to nurse ratio do you often see? And this was a question from Meredith Lewis, and she stated that I either have 1 or 2 patients depending on the patient acuity. So thank you for that. For all 3 of you, when it comes to students prior to starting college, volunteering, getting internships, gaining experiences, and landing a job, are there certain skills that have helped you transition from before your career, or any sort of groups or sort of hobbies that you would say that this is something that they should look at?
I can answer that. So in high school, I didn't have that many opportunities for research as there are now. If you just, there's plenty of opportunities now. But one of the things that I did was candy stripe. And I went to a hospital. I asked like the receptionist or somebody, hey, do you guys have a candy stripe program? And do you guys have candy stripers that can come into your labs? And so, yes, essentially, yes. So asking that question, remember, ask questions. So asking, hey, is there an opportunity here? Is a good way. And something that I would attribute to my success in general is my violin playing. So I started playing the violin when I was, I want to say 7 or 8. And I played it all the way up till college. And it really helped focus me on something that I was interested in. And there are plenty of studies that show that playing an instrument can actually help enrich your research skills. And so that was something that I did, just for my own leisure that ended up working out.
Thank you, Dr. George. And Meredith, is there something that you would recommend?
So I did, when I was in high school, I went to the Academies of Loudon program, and to figure out if nursing was something I wanted to do before I went to college. So if your public school program has something like that, it helped me a lot personally. But also, like Dr. George just said, volunteering in a hospital, because sometimes it looks a lot different than people think it does. So I think just being able to see it through that lens is always helpful. And then I think that what has helped me the most is, like Dr. George said, ask questions. Science is a naturally inquisitive field. No one is ever going to fault you for having a question. I feel like we love it. And there are no silly questions. So like in my day-to-day, I last week had a transfusion reaction and had to take the blood back to the blood bank. And I was like, what are you going to do with it? What happens now? Because I don't get told that part. So just always ask questions. Don't be afraid to look silly. I feel like when you're young, and that is a fear that I had, and it's not, just go for it. And then I think, volunteering and seeing in a practical application if that's something you would be interested in is helpful.
Perfect. Thank you. And lastly, Katie, any words of wisdom or opportunities that you would recommend to these young folk?
Sure. One thing that I struggled with was public speaking. I was terrified to have people look at me for a long period of time. And so even if I knew exactly what I was talking about and, I knew the content back and forth, I really would have preferred not to do that. So I talked with my bosses, which again, yes, I'd like to mirror your, mentors are so important because they can give you opportunities that challenge you to grow. So I was given the opportunity to train, 150 people at our company for about 3 years on different aspects of quality and compliance. And that's part of, what I do today. And also I'm much more comfortable in front of a crowd and it was really just because I talked to my boss and said, I have a fear. And she said, here's, let's just get you a lot of practice. And that's how you overcome your fear. And also I don't present perfectly, but that's part of it. You learn how to laugh when things don't go right.
Those are great words of wisdom. I have one other question for you, Katie. Can you speak about your time at the Research Science Institute?
Sure. Did you have a particular question about it?
No, I think that it would be really great if these students were just to hear a firsthand account. I think they could really learn from that.
Well, it was a wonderful experience. It was one of the first years at the MIT campus, so there was a lot of excitement about that. I worked at the BASF Bioresearch Corporation, and I worked with genes. I think I remember extracting genes and, running gels. And I think we were trying to discover a way to make purple roses. It was a while ago, so I don't exactly remember what, but I remember learning a lot of wonderful laboratory techniques that helped me, get my job at Burroughs Wellcome as well. And then also just, living for 6 weeks with other students with similar aspirations and interests, and also things that you've never thought of before in your life, because we were from all over, the country and all over the world. It was just a wonderful experience, and I can't say enough good things about it.
Thank you so much for speaking about it. For any student that's watching, I think it's a great opportunity and it'll be very beneficial for you. And you could also make some purple roses as well. Thank you again to Meredith, Jasmine, and Katie. Thank you also for our attendees for joining us. CEE appreciates all of our sponsors, including the Claude Moore Charitable Foundation. CEE supporters allow the center to host these events and provide other cost-free STEM educational opportunities. You will be notified when this recording is available on YouTube. Feel free to contact CEE's Manager of Executive Projects, Catherine Kai, with any questions or comments. And thank you, everyone.